So in the previous class, we were discussing about the cholinergic transmission, especially the production of acetylcholine, synthesis of acetylcholine, then the release of the acetylcholine, then destruction of acetylcholine, then its metabolism, followed by the action of the acetylcholine on the post and presynaptic receptors, then finally destruction of the acetylcholine. So a few of the important terminologies we have to know, especially the release of acetylcholine and other neurotransmitters by exocytosis through the pre-junctional membrane is inhibited by the Clostridium toxins. So whenever the Clostridium botani, Clostridium tetani and many other toxins are released by the respective bacteria, they are going to act on the exocytosis and going to inhibit the release of the neurotransmitter acetylcholine from the site of its action. And some of the most potent toxins are produced by the microbe, especially the Clostridium toxins inhibit synaptobrevin and related proteins in the nerve terminal. So, synaptobrevin, synaptotagmin, and many other RAP3, etc. are the proteins which are the docking complex, means they are going to fuse the vesicles with each other and going to release the neurotransmitter. So this process is blocked by the Clostridium toxins and there is no exocytosis of the neurotransmitter. Hence, what happens? There is no action of the acetylcholine and the paralysis occurs. And internalization in turn enables the small subunit to proteolyze synaptobrevin or the other target proteins. The internalization, especially the synaptobrevin, synaptotagmin, and all these are known as the what you call as the docking complex. So docking complex will be inhibited by most of the botulinum toxins, and botulinum toxin A binds to cholinergic motor nerve terminals, giving rise to flaccid paralysis. And the tetanus toxide, you know, it's a very popular disease, tetanus, Clostridium tetani. It's released from the Clostridium tetani, which causes the tetanus. It binds to and enters spinal neurons where it blocks glycine release and cause spastic paralysis. This tetanus toxin is nothing to do with the adrenergic nervous system, but it is entering the spinal nerves and binding the glycine residue. Glycine is a inhibitory neurotransmitter. So inhibition of the inhibitory neurotransmitter causes excitation and causes spastic paralysis. So once internalized, the larger functions as a selective zinc dependent protease and hydrolyzes the target protein. The toxin from another thing very interesting is the black widow spider venom. It's also known as alpha latrotoxin. So promotes massive vesicle release, presumably by binding to the neurexins on the neuronal membrane. Usually this black widow spider, the name arrived, especially the black widow spider, it secretes the alpha latrotoxin and promotes the massive vesicular release, especially of the cholinergic neurotransmitter, especially acetylcholine, and it binds to the neurexins on the neuronal membrane. Because of this thing, the acetylcholine is not released and causes the paralysis. So this is a black widow spider. Black widow spider, you can see this is very common in case of the African forests, and many times if the human beings are there, means it's going to bite, especially the human beings. Then what happens is there is the paralysis because the black widow spider is containing the toxin known as the alpha latrotoxin, and it's going to bind with the neurexin or syntaxin. Then once again the acetylcholine is not released, which are going to cause the paralysis. So here you can see this black widow spider. Why it is the name black widow spider is it is going to eat or consume the male spider. That's why it is known as the black widow spider. So that's name arrived. So this is containing a very potent toxin known as the alpha laterotoxin. And many times the persons who wander in the forest will have a severe bite of this particular widow spider and get paralyzed. And here also you can see that the black widow spider is biting a human and causes the paralysis of different nature or different degrees depending upon the quantity of the venom and the bite of the particular spider it is going to have. Especially even though it's a very small bite, it causes severe what you call as the reactions and also the paralysis in case of the human beings. And following section, see here there are few terminologies are there, please learn them. So the following section and degeneration of the motor nerve to skeletal muscles of or the postganglionic fibers to autonomic effector, there is a marked reduction in the threshold doses of the transmitter and certain other drugs required to elicit response. So what happens if you cut the nerves which are supplying to motor nerve and also the skeletal muscles and also the postganglionic fibers to autonomic effectors, the reduction in the threshold to the transmitter it causes, or else the tissue becomes very sensitive to the released neurotransmitter. This is known as denervation supersensitivity. Already it is told, denervation supersensitivity. Whenever a nerve supplied to a muscle is cut and the muscle become very much responsive to a small quanta of the neurotransmitter, and this is known as the denervation supersensitivity. In skeletal muscle, this damage is accompanied by a spread of the receptor molecules from end plate region to the adjacent portions of the sarcoplasmic membrane, which eventually knows the entire muscle surface. So what happens whenever the nerve is cut, which is supplied to a muscle fiber, then what happens, the receptor molecules from end plate region, they spread to the adjacent portions of the sarcoplasmic membrane, hence make the tissue very sensitive. Hence innervation represses the expression of receptor gene by the nuclei that lie the extra junctional region of the muscle fiber. So the receptors, suppose there are one or two receptors, just imagine, then they will be spread to the adjacent sarcoplasmic membrane and makes the tissue very sensitive to this neurotransmitter, and this is known as the denervation supersensitivity. Now let us deal about the adrenergic neurotransmitter. Here the synthesis, storage and release of the catecholamines occurs, and also the destruction of the catecholamines as usual in the all other neurotransmitter transmission like cholinergic transmission. Also we have learned the synthesis, how the adrenaline or norepinephrine are synthesized, how they are stored, and what is the mechanism of the release and the initiation of post-junctional activity, then destruction of the neurotransmitter. All these processes are coming in the adrenergic transmission. The norepinephrine is the transmitter of most sympathetic postganglionic fibers and certain tracts in the central nervous system. It's a very well established factor. Norepinephrine is one of the important neurotransmitter in most of the sympathetic postganglionic fibers and also in the central nervous system. Whereas the dopamine is the predominant transmitter in the mammalian extrapyramidal system or brain, and several mesocortical and mesolimbic neuronal pathways, because the Norepinephrine is converted to dopa to norepinephrine is formed. So dopamine is one of the important neurotransmitter, especially in the central nervous system. Whereas the epinephrine, the major hormone of the adrenal medulla. See, the three congeners are they are known as the catecholamines. Because why the name catecholamine is coming? Because they have got a very specific ring known as the chemical ring known as the catechol. So the norepinephrine, dopamine and epinephrine are the three important catecholamines. The first one, norepinephrine, is a neurotransmitter in the postganglionic fibers and many muscles, etc. And the dopamine, especially, it is working on the central nervous system, whereas the epinephrine is the major hormone of the adrenal medulla. Very simply, you can understand all these things. Yes, this is a very important what you call as the structure, and we can remember this one for the synthesis, then the formation of the epinephrine, whereas the norepinephrine. So here, this is the structure of the what you call as this is the structure of the tyrosine. Tyrosine. Please go step by step. This is a hydroxyl moiety. This is CH2, CHCOOH, and amide. So this is side chain is amide. So this is known as the tyrosine. Tyrosine. So this tyrosine is uptaken from the exoplasm into the from the surrounding peripheral tissue. So the enzyme here which converts tyrosine to dopa is tyrosine 3 monooxygenase. The enzyme is tyrosine 3. In the place 3, third position, it is called as monooxygenase, means one oxygen moiety will be attached by the help of this tyrosine 3 monooxygenase in the third position of this catechol ring. This entire ring is known as the catechol ring. So they are the catechol amines, especially dopa, dopamine, norepinephrine. All these are the catechol amines. You might have understood why the name catecholamine came. Then the tyrosine is converted to dopa with the help of tyrosine hydroxylase. The other name of tyrosine 3 monooxygenase is tyrosine hydroxylase, and the catalysts are tetrahydroperine. So this is the catalyzing protein. See here, one OH molecule is added and then becomes the dopa. Dopa is going to become dopamine with the help of aromatic L-amino acid decarboxylase. The dopa is going to convert it to dopamine, or it is also known as aromatic L-amino acid decarboxylation process occurs here. See, please see, this is a dopamine, and the change what happens is instead of this COOH, it is removed here and one hydrogen moiety is attached. So the structure is dopamine. Dopa, one amine group is added, or H2 molecule is added, and the acid moiety COOH moiety is removed. So dopa to dopamine, and dopamine is going to converted from dopamine to norepinephrine with the help of the enzyme dopamine beta hydroxylase. Dopamine beta hydroxylase. In the beta position of the dopamine, hydroxylation occurs. So this is alpha and this is beta. Here you can see that one OH group is added to from the dopamine to norepinephrine. So the enzyme which converts norepinephrine to epinephrine is phenylethylamine N-methyl transferase. Here you can see the phenylethylamine N-methyl transferase, especially the amide group is present instead of the one H group, one CH3 is one methylation is going to occur here. See, the norepinephrine is methylated to form epinephrine, especially in the adrenal cortex. And once again, to go through tyrosine is converted to dopa, dopa to dopamine, dopamine to norepinephrine, norepinephrine to epinephrine. The enzymes involved are tyrosine 3 hydroxylase or monooxygenase, or it's also known as tyrosine hydroxylase in the second. So here in this group, one OH moiety is added, then dopa to dopamine aromatic L amino acid decarboxylase, or the pyridoxal phosphate is the what you call as the catalyst here, which makes the enzyme to work faster. And dopamine to norepinephrine is uh, catalyzed by the enzyme dopamine beta hydroxylase, and the ascorbate is the catalyst. And the norepinephrine is converted to epinephrine with the enzyme phenylethylamine N-methyl transferase. So it is very easy to remember if you go through the chemical changes, series of the chemical changes occur. First, the one hydroxylase group is added, then instead of this carboxyl COOH moiety, one hydrogen moiety is added in the beta position. Then this with the help of dopamine beta hydroxylase in this beta position, one hydroxyl moiety is added, and N-methyl transferase, phenylethylamine N-methyl transferase, norepinephrine is converted to epinephrine with the addition of one CH3 molecule. Hope this is very clear to you. And here, so many enzymes are also participating in the synthesis of the catecholamine varieties of the enzymes. The first enzyme that's going to participate is the tyrosine hydroxylase. Here it is present, tyrosine hydroxylase or tyrosine 3 monooxygenase is the enzyme, and it is going to its occurrence is widespread, and it is present in the most of the sympathetic nerve, and it is present in the cytoplasm especially, or it is also known as the exoplasm. And the cofactor requirement is tetrahydrobiopterin and oxygen and a ferrum moiety, and substrate specificity is very specific for L-tyrosine. So this is going to convert to tyrosine to dopa, and this is a rate limiting step, and inhibition can deplete norepinephrine. Usually if you inhibit this tyrosine hydroxylase, then there is depletion of norepinephrine. And the second enzyme is aromatic L amino acid decarboxylase, means here it is present aromatic L amino acid decarboxylase. Here the decarboxylation occurs. COH is this decarboxylation, especially the dopamine to dopa to dopamine, it's going to convert. This carboxyl moiety is removed and hydrogen moiety is added. That's decarboxylase. And widespread sympathetic its presence, and it's also present in the cytoplasm. The pyridoxal phosphate is a cofactor here. Here you can know the pyridoxal phosphate. The pyridoxal phosphate is the cofactor which is going to catalyze this particular amino acid aromatic L amino acid decarboxylase, and it's a non-specific in action, and inhibition does not alter the tissue norepinephrine or epinephrine. That's as it is not specific, and it is not going to change anything if you inhibit that particular enzyme. And the third enzyme is dopamine beta hydroxylase. In the beta position, the hydroxylation is going to be caused by this dopamine to norepinephrine is going to be converted, and the widespread, and it is present in the sympathetic nerves, and this it is present in the synaptic vesicles. Please remember that dopamine beta hydroxylase enzyme is present in the synaptic vesicles, and the cofactor requirement is ascorbic acid and oxygen, and which contains the copper. Here you can see that the ascorbate is present, or ascorbate present with that as a cofactor. And once again, the action is very non-specific. It will act on the many type of the dopa to dopamine, and inhibition can decrease to norepinephrine and epinephrine release. Even though it's a non-specific, its inhibition decrease the norepinephrine and epinephrine levels. The another thing is the phenylethylamine. This is the last enzyme which is present in the last step, norepinephrine to epinephrine. This is present in the adrenal gland, because norepinephrine is converted to epinephrine because it's methylated there, then it's going to stored in the adrenal gland, The distribution of this enzyme is in the cytoplasm, especially the cytoplasmic one, and the cofactor present is the S-adenosyl methionine because it's a CH3 donor. Phenylethylamine N-methyl transferase causes one methyl group to be donated to the norepinephrine, and it is converted to epinephrine and stored in the adrenal gland. And it's also a non-specific enzyme, and inhibition can decrease adrenal what you call as the norepinephrine regulated by glucocorticoids. Especially this is regulated by glucocorticoid. Whenever there is more and more glucocorticoids are released, then more and more epinephrine is released from the adrenal gland. In the adrenal medulla, so the catecholamines are stored in chromaffin granules. It's known as chromaffin granules, and these vesicles contain approximately high concentration of the catecholamines, approximately 21% of the dry matter, especially the adrenal medulla. If you cut open it and dry, then 21% of it is containing the catecholamine. And apart from this one, the ascorbic acid is present, and the adenosine triphosphate is present, and the chromogranins are present, whereas the enzyme which converts this norepinephrine to epinephrine and also dopa to dopamine is dopamine beta hydroxylase DBH is present. And apart from this one, enkephalins and neuropeptide Y, it is these are the contents of the adrenal medulla, whereas the adrenal cortex do contain the corticosteroids, and they are very important for the release of the catecholamines from adrenal medulla. So the entire combination of the adrenal medulla is 21% is containing the catecholamines, the ascorbic acid, remaining is ascorbic acid, ATP, chromogranins, and DBH, dopamine beta hydroxylase, and enkephalins, and also the neuropeptide Y. See, this 5-HT, 5-hydroxytryptamine can be produced by aromatic L-amino acid decarboxylase or dopa decarboxylase from 5-hydroxy N-tryptophan. So 5-hydroxy-L-tryptophan is just like the tyrosine, and it's taken into the exoplasmic membranes. Then it is converted to 5-hydroxytryptamine. And dopamine beta hydroxylase also converts dopa to dopamine, and methyl dopa is converted to alpha methyl dopa, which in turn is converted by dopamine beta hydroxylase to a false transmitter because it's a what you call as a substrate for the dopamine beta hydroxylase enzyme, and the false transmitter is alpha methyl norepinephrine. Once again, another methyl group will be added to the norepinephrine, and it's called as the what you call as the alpha methyl dopamine, and dopamine beta hydroxylase is the enzyme which is involved, and this is a false transmitter theory or false transmitter. The hydroxylation of tyrosine generally is regarded as the rate limiting step. That's the tyrosine to dopa, conversion of tyrosine to dopa. With that, it's a rate limiting step in the biosynthesis of catecholamines, and tyrosine hydroxylase is activated following stimulation of the adrenergic nerves or adrenal medulla. So, whenever a neurotransmitter to be released, there should be an enzyme which is very specific to that of the particular step. And this tyrosine hydroxylase is a very specific enzyme which converts tyrosine to dopa, and it's activated following the stimulation of adrenergic nerve endings or adrenal medulla by corticosteroids. And this is a very important step here, and you can learn this particular step. Like this is a sympathetic nerve. Here it's a sympathetic nerve, and this is a effector cell. Here this is a effector cell, and the tyrosine is taken up from the periphery into the cytoplasm of the sympathetic nerve, and tyrosine is converted to dopa to dopamine, and dopamine to norepinephrine. It's converted to norepinephrine. So norepinephrine is going to be taken up, especially in the vesicles, vesicular uptake. Then it is going to have the fusion of many vesicles. Then it is going to be released by the arrival of the action potential. Then the released norepinephrine may act on alpha type of the receptor or beta type of the receptor, especially in the neuroeffector junction. And apart from this one, it may also act on the presynaptic alpha receptor, and the going to change. Then here you can see that the monoamine oxidase, that's the first enzyme, is going to act on the norepinephrine and going to break down it to a metabolite, inactive metabolite, which is present in the sympathetic nerve varicosity, that is the exoplasm. And these are all the uptake ones. Norepinephrine is what you call as gives a feedback mechanism. If the concentration of norepinephrine is going to be increased, then it gives a feedback mechanism, negative feedback mechanism, so that the further metabolism of tyrosine will not occur. Usually, the norepinephrine is going to be released after exocytosis, and there is a response beta or alpha receptors. Through alpha or beta receptor, there will be a response will occur after the post junctional activity. So this entire diagram explains the synthesis, the storage, the release of norepinephrine, and also initiation of the post junction activity, and also the metabolism of the norepinephrine. Here you can see this entire step with different letters. Yes, the enzymes that participate in the formation of norepinephrine are synthesized in the cell bodies of the neurons and are then transported along with their axon to their terminals. So these enzymes, especially dopamine beta hydroxylase and tyrosine hydroxylase, these enzymes are formed in the cell bodies of the neurons and then are transported along the nerve terminals. So in the course of the synthesis, the hydroxylation of tyrosine to dopa, and the decarboxylation of dopa to dopamine like takes place in the cytoplasm, or we can name it as the exoplasm. About half of the dopamine formed in the cytoplasm is then actively transported into the dopamine beta hydroxylase containing storage vesicles, where it is converted to norepinephrine. Here you can see the step that dopamine beta hydroxylase, this particular step you can see. Yeah, please remember this particular diagram, which is a very basic or fundamental to all the next processes, because it is very important to remember tyrosine converted to dopa with the help of the enzyme. First enzyme is tyrosine hydroxylase, TH, tyrosine hydroxylase, which causes one hydrogen moiety to be attached to the benzene ring, which is later the dopa decarboxylase or L-amino acid decarboxylase enzyme. It is an enzyme. Then dopa to dopamine is converted, because here this OH is removed and one hydrogen moiety is added. Then dopamine beta hydroxylase is enzyme which converts dopamine to norepinephrine. Dopamine beta hydroxylase, in the beta position one hydroxyl group is added, whereas the norepinephrine is going to be converted to epinephrine with the help of phenylethanolamine N-methyl transferase. This is the sequential structure. So usually the dopamine beta hydroxylase it is converted to norepinephrine, The remainder which escaped capture by the vesicle is deaminated to 3,4-dihydroxy phenyl acetic acid, or it's also known as DOPAC 3,4-dihydroxy phenyl acetic acid, and subsequently it is methylated in the O position to homovalenic acid, or it's also known as HVA. So this is about the metabolism of the norepinephrine. See, the uh, dopamine beta hydroxylase is going to convert norepinephrine to no, to dopa to norepinephrine, where it is converted to norepinephrine, whereas the remainder escaped captured by the vesicles, and it is deaminated by 3,4-dihydroxyphenylacetic acid. It's also known as DOPAC, and subsequently O-methylated to homovalenic acid. This is the metabolism pathway of the norepinephrine. The adrenal medulla has two distinct catecholamine containing cell types. One is with norepinephrine and those with primary epinephrine. So the adrenal medulla, it's a very good storage house of the norepinephrine and epinephrine. Both are there, and it is containing the two cell types, one with norepinephrine and primary epinephrine. The later cell population, especially the primary epinephrine, contains the enzymes phenylethanolamine N-methyl transferase. That's the enzyme which converts norepinephrine to epinephrine. So it is known as phenylethanolamine N-methyl transferase, because one methyl group is transferred to norepinephrine, and that norepinephrine becomes epinephrine. And in these cells, the norepinephrine formed in the granules leave the structure presumably by diffusion and is methylated in the cytoplasm to cause epinephrine, that you have very specifically seen in the diagrammatic representation. And then what happens? Epinephrine then re-enters the chromaffin granules, where it is stored until it is released. So epinephrine is stored in the chromaffin granules. Chroma means the colored granules, chromaffin granules, where it is stored until the release. The release of the epinephrine occurs after the stimulation of adrenal cortex and release of the corticosteroids. That was flight or fight reflex. So epinephrine accounts for approximately 80% of the catecholamines of the adrenal medulla, with norepinephrine making 20%. So the ratio of epinephrine to norepinephrine in adrenal medulla is epinephrine to norepinephrine 80% to 20%, 80 to 20. So that's why the tissue is made always responsive, and a massive epinephrine will be released from the storage site and acts on the different cells. So here you can see that three enzymes, these three catechols, DOPA, dopamine, and norepinephrine. These are the three catecholamines which are synthesized with the help of varieties of the enzymes and some of the substances. The tyrosine is uptaken into the exoplasm by the tyrosine beta hydroxylase enzyme, and it's converted to dopa to dopamine, and dopamine to norepinephrine, norepinephrine to epinephrine. So these are stored in the storage vesicles, especially in the presynaptic nerve terminals. So then they are going to be fused, and exocytosis occurs, and the neurotransmitter acts on the postsynaptic receptors. And you can see that the tyrosine is uptaken from the nerve endings. That's the rate limiting steps, which is acted by tyrosine hydroxylase enzyme. And the synthesis of norepinephrine depends on the size of the store available for release from the adrenal medulla, the level of glucocorticoids secreted by the adrenal cortex. So it's very interrelated. Whenever the glucocorticoids are secreted, they are going to act on the cells, especially the uh, enter these chromaffin cells, especially on the chromaffin cells, then they are going to be acted on the postsynaptic vesicle. And in adrenal medullary chromaffin cells, synthesis of phenylethanolamine N-methyl transferase, that's the last enzyme which is required to convert norepinephrine to epinephrine. The synthesis of this phenylethanolamine N-methyl transferase takes place. The activities of both tyrosine hydroxylase and dopamine beta hydroxylase increased in the adrenal medulla when secretion of glucocorticoid is secreted, because the glucocorticoids are the one which make lot of epinephrine to be released from adrenal medulla. So the synthesis of both the enzymes required for the synthesis of the norepinephrine and epinephrine, like tyrosine hydroxylase and dopamine beta hydroxylase, they are increased. And secretion of corticotrophin metabolize the hormones of both adrenal cortex. Predominantly the cortisol is present, cortisol and the medulla. The medulla contains the epinephrine, whereas the adrenal cortex contains the cortisol. So the cortisol is released into the bloodstream, then it's going to act on the receptors of the medulla and releases the epinephrine. They are highly interrelated. Here in this particular diagram, you can very clearly see that the tyrosine transporter is present and tyrosine is uptaken into the exoplasm, and then tyrosine is converted to L-dopa or dopa tyrosine hydroxylase, or the tyrosine hydroxylase is going to convert tyrosine to dopa. Dopa is converted to dopamine with the enzyme dopamine decarboxylase, and dopamine to norepinephrine is by dopamine beta hydroxylase is the enzyme. And so this is uptaken, and the 3,4 dihydroxymandelic acid will be formed after the action of monoamine oxidase on the norepinephrine, whereas the calcium, whenever this type of reactions are occurring, then exocytosis occurs due to the increased calcium content because of the voltage related calcium channels. Catechol O-methyl transferase and monoamine oxidase are the two important enzymes which are what you call as the destroy the norepinephrine, or especially the particular the destruction of norepinephrine or epinephrine or both. Catechol O-methyl transferase, it is converting norepinephrine to normetanephrine. So the monoamine oxidase, it acts on the norepinephrine and forms 3,4 beta hydroxy mandelic acid. Then this is converted to vinyl mandelic acid, or it's also known as VMA. Then they are all interchangeable, especially normetanephrine. Catechol O-methyl transferase acts on the norepinephrine to cause this particular normetanephrine. Normetanephrine is going to be converted to vinyl mandelic acid, and this is going to be excreted through urine. So this entire diagram is a very informative, because here it informs about the synthesis, the storage, then release, and the metabolism of the neurotransmitter norepinephrine or epinephrine, and also the post-junctional activity, because the norepinephrine is going to bind with either alpha or beta type of the receptors. Here the alpha or beta type of the receptors are present, and the metabolism of norepinephrine in the synaptic cleft occurs by catechol O-methyl transferase enzyme, whereas inside the cytoplasm or the exoplasm, monoamine oxidase is the very specific enzyme. So hope you understood this entire concept. The adrenal chromatin cells are developed entirely by steroid secreting cortical cells enveloped by. So the adrenal chromatin cells, especially in which this epinephrine is stored, it's surrounded by the glucocorticoid, the cortical secreting cortical cells, and termination of the effects of adrenergic impulses in most organ is due to the recapture or reuptake by the active transport of norepinephrine previously released into the extracellular fluid. See, whenever the stimulus come at the exoplasmic reticulum, then it is going to cause the exocytosis of the norepinephrine containing vesicles and a massive release of the norepinephrine occurs. But totally it's impossible, unlike the acetylcholinesterase, to break down norepinephrine to the metabolite. Here, an important step is there, that's known as the reuptake of the norepinephrine with active transport with the help of the energy. It is going to be uptaken from the exoplasmic terminals, and two processes are there uptake. So this is known as the uptake 1 and uptake 2. They are known as the uptake 1 and uptake 2. The thing is uptake across the exoplasmic membrane from extracellular fluid to the cytoplasm, especially the neuroplasm. So whenever the action potential comes and a massive norepinephrine is released into the from the storage vesicle inside the synaptic cleft, there even though the catechol O-methyl transferase enzyme is present, just like acetylcholinesterase, it is not so strong and very specific to that of the acetylcholinesterase. So, the huge quantity of the norepinephrine released is reuptaken back into the cytoplasm. Then, uptaken from cytoplasm into the storage vesicles, especially the vesicles. The first one is uptaken into the exoplasmic membrane from the extracellular fluid to the cytoplasm. That's the uptake 1, whereas the uptake from cytoplasm into the storage is known as uptake 2. And the storage of the catecholamines. So as usual, the norepinephrine and epinephrine are stored in vesicles to ensure their regulated release. Why the storage is required? Because whenever the action potential arrives, it is not easy to synthesize the norepinephrine or epinephrine so fastly. So they need to be stored for the release whenever the action potential comes or whenever there is requirement of the neurotransmitter. And this storage decreases intraneuronal metabolism of the transmitters as well as their leakage outside the cell. The thing is, when they are stored in the storage vesicle, it is having the barrier so that the enzymes should not act on them. And for the uptake, it is uptake 1 of the catecholamines is a ATP dependent process, so that the chromaffin granules, especially in case of the adrenal medulla, the uptake of catecholamines and ATP into isolated chromaffin granules driven by pH and potential gradients are established by an ATP dependent proton translocase. So here two neurotransmitters are there, or catecholamines. One is norepinephrine and epinephrine. Norepinephrine is the one which is going to be stored in the storage vesicles in the nerve endings, whereas the epinephrine is going to be stored in chromaffin granules. For every molecule of amine take-up, two hydrogen ions are extruded out. So for every single molecule of the norepinephrine or epinephrine, whatever it may be, taken up, two hydrogen ions are extruded out. And monoamine transporters are relatively promiscuous and capable of transporting dopamine, norepinephrine, epinephrine, and serotonin. All these neurotransmitters are being transported by the monoamine transporters, and they are not so specific. They are going to transport each and every enzyme, just like the monoamine transporter 1, 2, 3, etc. And dopamine, norepinephrine, epinephrine, and serotonin. All these neurotransmitters are being acted by the monoamine transporters. So, reserpine is a drug that inhibits monoamine transport into vesicles, which ultimately leads to depletion of catecholamines from sympathetic nerve endings and in the brain. So, reserpine is a drug obtained from the plant Rauwolfia serpentina, and it is the antihypertensive drug. So, what is the mechanism of reserpine is reserpine is going to inhibit monoamine transporter into the vesicles, which ultimately leads to depletion of the catecholamines from sympathetic nerve endings and in the brain. So because of both of this combined effect, there is decreased blood pressure occurs, especially if you consume the reserpine. The amine transport system across exoplasmic membrane is sodium dependent. So the amine transport system in the exoplasmic membrane is sodium dependent, and this is blocked selectively by a number of drugs including cocaine and tricyclic antidepressants such as imipramine. So cocaine. So cocaine is an addictive drug. It is going to block the transportation of the norepinephrine from the exoplasmic membrane. So, and apart from this one, the tricyclic antidepressants such as imipramine, desipramine, and tranylcypromine, these are going to this bind with the transporter system and prevent the transportation of the norepinephrine into the cytoplasm. And high affinity transporters have been identified now, for example, for dopamine, norepinephrine, and serotonin, and a variety of the amino acid transporters. And why we are learning, especially all these enzymes and the precursors, and so many is so to find a pharmacological agent which can intervene with that of this normal physiological process so that we can get a required pharmacological and therapeutic effect. So that's how the purpose of learning this particular what you call as the metabolism, storage, etc. in a very specific manner. Ephedrine, amphetamine, and tyramine are indirectly acting sympathomimetic amines. Ephedrine, amphetamine, and tyramine, which are indirectly acting sympathomimetic amines, act by displacing norepinephrine from nerve endings, binding sites to the extracellular fluid. So what happens this whenever we administer ephedrine, amphetamine, tyramine to the living organism, these are known as the indirectly acting sympathomimetic amines. So what they are going to do, they are going to displace the norepinephrine from the nerve ending binding sites to the extracellular fluid, and the released endogenous transmitter then acts at the receptor sites of the effector cells, and this is known as as such the agents are substrate for uptake 1. Uptake 1 means transportation of the catecholamines from the exoplasm to the storage vesicles. That's known as the uptake 1. And these actions of indirectly acting sympathomimetic amines are associated with the phenomenon of the tachyphylaxis. Tachyphylaxis means as the quantity of the neurotransmitter is going to be increased, instead of increasing the response is going to be decreased. That is known as the tachyphylaxis. And repeated administration of tyramine, ephedrine, amphetamine, and tyramine, which displace the neurotransmitter from the binding site, causes the rapidly decreasing effectiveness whereas the repeated administration of norepinephrine does not reduce effectiveness, and in fact it reverses the tachyphylaxis to tyramine. So the tyramine, which is a what you call as the indirectly acting sympathomimetic amines, they are going to act or displace the norepinephrine from the binding site. Whereas the uptake 2 is not inhibited by the imipramine or cocaine, because uptake 2 is happening from the exoplasmic to storage vesicle. Uptake 1 from the what you call as the intracellular fluid to the exoplasm, that transport, this is inhibited by the ephedrine, amphetamine and tyramine, whereas and also the cocaine, and uptake 2 is not inhibited by imipramine and cocaine. Then the release of the acetylcholine, not the catecholamines, in the adrenal medulla, because adrenal medulla do contain lot of the norepinephrine in the bromophenol granules. The triggering event is the liberation of acetylcholine by the preganglionic fibers and its interaction with nicotinic type of the receptor on chromaffin cells to produce. See, the nicotinic type of the receptors are present on the chromaffin cells. Nicotinic acetylcholine or muscarinic type of the nicotinic type of the receptors are present on the chromaffin cells, and the liberation of acetylcholine by the stimulation of preganglionic fibers will going to cause a localized depolarization, entrance of calcium into these cells, which extrusion by exocytosis of the granular contents, means the granular content is having epinephrine, ATP, neuroactive peptides or their precursors, and chromogranules. So these are all the content of the chromaffin granules along with the epinephrine. So all these are going to be released at the same time along with the epinephrine and going to act on the receptor. So what happens? The subsequent events are the CS whenever this type of this content of chromaffin cells along with the other additional things, especially the calcium entry occurs, localized depolarization, and all the other neuroactive peptides or their precursor chromogranins and dopamine beta hydroxyls. All these are simultaneously released. So what happens is there will be an increase in dopamine beta hydroxylase and chromogranin release in the circulation. So adrenergic fibers can sustain the output of norepinephrine during prolonged periods of stimulation without exhausting their reserve supply, provided synthesis and uptake of the neurotransmitter are unimpaired. Means the adrenergic fibers can sustain the output of norepinephrine during a prolonged period of stimulation. Continuously, if you are stimulating the adrenergic fiber, then what happens is without exhausting their reserve supply, they are provided with synthesis and uptake of the transmitter. They are not impaired. And there will be activation of tyrosine hydroxylase and dopamine beta hydroxylase, DBH, and dopamine to norepinephrine is going to be catalyzed. And termination of the action of the catecholamine occurs. Ultimately, the action of the norepinephrine and epinephrine are terminated by the number 1, reuptake into the nerve terminals. Maybe uptake 1 and uptake 2, reuptake of the nerve terminals, and dilution by diffusion out of the junctional cleft and uptake at extraneuronal sites. See, the first one is reuptake into the nerve terminals. Second one is dilution of that particular neurotransmitter vicinity by diffusion to the adjacent or junctional cleft and uptake at extraneuronal sites. Then the third one is the metabolic transformation. That means the norepinephrine or epinephrine, they are converted to their respective metabolites like just like norepinephrine and methanephrine. And here two important enzymes are having the metabolism of the catecholamines. The number one is MAO or monoamine oxidase is the important enzyme, followed by catechol O methyl transferase, is also known as COMT. So these two enzymes are very important in the metabolism of the catecholamines. However, it is evident that a powerful enzymatic pathway such as the provided by acetylcholinesterase is absent from the adrenergic nervous system. So whatever may be, these two enzymes are present, but they are not as strong as acetylcholinesterase, which is present in the cholinergic nervous system. And these are not so strong when compared to the acetylcholinesterase, which is going to metabolize the acetylcholine into acetic acid and choline. So in a flash-like manner, that's very strong enzyme, whereas the monoamine oxidase or catechol O-methyl transferase are there, but they are not so effective as acetylcholine esterase. And the norepinephrine released in nerve terminal is metabolized by the monoamine oxidase. Norepinephrine released in nerve terminal is going to is metabolized to it is what you call as it's metabolized to the water soluble moiety and going to be secreted out. And another enzyme, that's the catechol O-methyl transferase, which converts norepinephrine to which is going to catechol O-methyl transferase is converting these catecholamines to the inactive substances like homovanillinic acids, etc., which are going to be secreted in urine, some metabolized. This is present very specifically in the liver, plays a major role in the metabolism of endogenous circulating and administered catecholamines. So, if you exogenously administer certain norepinephrine or epinephrine, then the catechol O-methyl transferase, it is going to be activated, then it is going to decrease the concentration of the enzymes, especially the norepinephrine. And both monoamine oxidase and catechol O-methyl transferase are widely distributed throughout the body, including inside the brain. And highest concentration of each are present in the liver and in the kidney, especially the monoamine oxidase and catechol O-methyl transferase. Both are present in the liver and also in the kidney, and also they are also present especially in the nerve endings, in the synaptic cleft. Catechol O-methyl transferase or COMT plays minor role in adrenergic metabolism. So even though it's an enzyme which is present in the adrenergic nervous system, its role is minor, unlike the acetylcholinesterase, which is one of the major what you call as the enzyme which destroys the acetylcholine within a fraction of seconds. It is not like that. It is not so important. And two different isoenzymes of monoamine oxidase are found in the central nervous system and in the peripheral nervous system. So, subtypes of the monoamine oxidases are present and subfamilies are also present. Epinephrine and norepinephrine are methylated by catechol O-methyl transferase to metanephrine and normetanephrine respectively. Then they are going to be converted to some water soluble moieties going to be secreted in the urine. That's how the metabolism occurs. So here this is a very specific chart. One has to remember this particular chart. Here you can see that the norepinephrine is acted by monoamine oxidase and going to be see here monoamine oxidase is going to act on norepinephrine and it is going to convert it to DOP, that's the short form of dihydroxyphenyl So 3,4-dihydroxyphenyl ethylene glycol. So this is dope gal, and the monoamine oxidase, especially the epinephrine, is also going to cause the DOPGOL. Whereas the norepinephrine is acted by catechol O-methyl transferase to catechol O-methyl transferase, and the methylation occurs here, and it is going to be converted to normetanephrine. Whereas the epinephrine, with the presence of catechol O-methyl transferase, it's going to metanephrine. Whereas the normetanephrine and metanephrine both are acted by the monoamine oxidase to form the MOPGAL, MOPGAL, and both of these things, they are going to aldehyde reductase and aldehyde dehydrogenase to MOPEG, and the dopeg, which is caused by the aldehyde reductase enzyme to cause the dopeg, and ultimately they are going to be cause this vinyl mandelic acid or the VMA, and going to be secreted in the urine. So this is the particulars, the pathway how the metabolism of norepinephrine and epinephrine occurs. Unlike that of the acetylcholine esterase, two enzymes are present. Of course, the monoamine oxidase enzyme is somehow more potent than that of the catechol O-methyl transferase, because the catechol O-methyl transferase converts the catecholamines to metanephrine, and metanephrine is actioned by the monoamine oxidase, and MOPGAL is going to cause, and the vinyl mandelic acid and MOPEG are going to be formed. So please remember this particular chart. So norepinephrine is initially deaminated by monoamine oxidase enzyme to 3,4-dihydroxyphenyl glycoaldehyde, 3,4-dihydroxyphenyl glycoaldehyde. And then this aldehyde, especially please see here, 3,4-dihydroxy monoaldehyde, 3,4-dihydroxy monoaldehyde. This is reduced to the glycol 3,4-dihydroxyphenyl ethylene glycol, or it's also known as DOPEG, DOPEG. This is DOPEG. Then to form then later this is acted by aldehyde dehydrogenase to form 3,4-dihydroxy mandelic acid, or it's also known as the DOMA, 3,4-dihydroxy mandelic acid, or also the DOMA. Then this is a generally but incorrectly called as vinyl mandelic acid, or VMA, is the major metabolite of the catecholamines. This is ultimately excreted in the urine. It is made as more polar and water soluble. The corresponding product of the metabolic degradation of the dopamine, which contains no hydroxyl group, the side chain, is homovalenic acid, HVA, and this is going to be secreted in the urine. And the measurement of the catecholamines and their metabolites in the blood and urine is useful in the diagnosis of pheochromocytoma, a catecholamine secreting tumor of the adrenal medulla. So suppose there is a tumor like pheochromocytoma, then the metabolite concentration will be measured in blood. Then it is an indicator of the tumor, and the inhibitors of MAO like pargelin and nialamide can cause increase in the concentration of norepinephrine, dopamine, and 5-hydroxytryptamine in the brain and other tissues, accompanied by varieties of the pharmacological effects. Whereas no striking pharmacological action can be attributed to inhibition of COMT. That means it's not an important enzyme. Just if you compare to the acetylcholine esterase enzyme, so it is present but not so important, especially in case of adrenergic nervous system. The enzymatic degradation is not so important, whereas the uptake and other process are so more important for the metabolism of the neurotransmitter. So we will deal the classification of the adrenergic receptor in tomorrow's class.